Hi everyone, we're Group 11 and we did our project on Ethidium bromide and other intercalating dyes. Let's jump right into it and have Brandon start us off with a brief history of Ethidium bromide itself.
The use of Ethidium bromide had essentially been around since the late 1940s, but it had a more medicinal purpose. Back then, it was originally a compound known as dimidium bromide, which had a structure that was mostly similar to the compound we know today, but it was used as an antibiotic. This antibiotic was used to treat infections that were running rampant through cattle and pigs in Ethiopia. It wasn't until the late 1950s that scientists conducting research on DNA structure discovered that Ethidium bromide could easily move around the double helix structure and lock on, a phenomenon known as intercalation, which will be touched upon later in this video. By doing so, it inhibited the reproduction of viruses and bacteria, but it also made the DNA glow bright orange when placed in ultraviolet light. Despite the possibilities that this discovery introduced, it wasn't until 1972 that it was used to its full potential. A Dutch physician named Piet Borst was working in his lab when a centrifuge broke down. This prevented him from conducting his research into DNA structure. He decided to implement a technique called gel electrophoresis, which separates DNA by size by running the sample through a gel using an electric current. This separated the DNA, but he still had trouble visualizing the distinct bands of his sample. That's when he remembered the fluorescent reaction that occurred between Ethidium bromide and DNA when they come into contact with each other. He decided to stain some of the gel with Ethidium bromide and watched as the individual bands glowed bright orange. Due to the success of this experiment, Ethidium bromide went on to become the standard for all DNA staining practices. And now, Gia will explain the mechanism behind this reaction.
Thank you, Brandon. So, how does Ethidium bromide work? You can see here that it has 4 planar aromatic rings. The entire molecule can be inserted between 2 adjacent base pairs of double-stranded DNA. This is known as intercalation, and it is driven by the enthalpically favorable free energy change from van der Waals pi stacking between the aromatic systems of the dye molecule and the base pairs. During this event, negative unwinding of the helix occurs by 26 degrees from puckering and changes in torsional angles of the bonds within DNA. This increases the distance between base pairs such that the dye compound can be wedged into a groove. In most conditions, the minor groove is preferred since binding is very weak at the major groove. All this occurs nearly instantaneously, within a millisecond. Additionally, the polycyclic aromatic system in Ethidium bromide and other intercalating dyes act as the chromophore, or the functional unit responsible for perceived color. If you can recall from organic chemistry, a more extensive conjugated pi electron system results in a smaller HOMO-LUMO energy difference and therefore a longer absorbed wavelength for excitation to occur. For Ethidium bromide, a highly conjugated molecule, the excitation peak, or where the maximum light energy is given off, occurs at 300 nanometers. At this wavelength, an Ethidium bromide stained sample would produce an orange glow when placed under ultraviolet light, which exists in the range from 200 nanometers to 400 nanometers. Next up, we have Zoe to go over some of the other dyes that have emerged since the discovery of Ethidium bromide.
Let's talk about other intercalating dyes. Many exist, all functioning by similar mechanisms of binding. Simple intercalators are similar in structure and function to Ethidium bromide. One example would be propydium iodide. Propydium iodide is almost identical to Ethidium bromide. Note the 3 fused aromatic rings at the core of the structure. This is known as a phenanthronine ring, and thus these dyes are known as phenanthronine dyes. The distinguishing feature of this dye is the quaternary amine. The ionic property of this additional group allows for greater solvation. This means more water molecules shed upon binding and a greater relative increase in fluorescence. Ethidium monoazide is very similar in structure to Ethidium bromide, with the exception of the azide substituent rather than the amino. Photolysis converts azide to nitrine. The nitrine group can initiate addition reactions. Having already intercalated with DNA, the nitrine binds immediately to DNA by a covalent attachment. An important feature of this family of dyes is that they are cell impermeant, meaning that they cannot cross through the cell membrane due to the presence of the hydrophobic tails. In reality, these interactions are far more complex, but generally relate to the charge of the molecule. There are cell-permeant members of this family, however. For example, dihydroethidium. This molecule is almost identical to Ethidium bromide, with the exception of the hybridization of the carbon with the phenol substituent. The neutrality of this molecule allows it to cross the cell membrane, where it exhibits blue fluorescence in the cytosol. The net neutrality of this molecule allows it to cross the cell membrane, where it exhibits blue fluorescence in the cytosol. The molecule can be oxidized to Ethidium, at which point it can intercalate and exhibit red fluorescence. Dyes of the phenanthronine family also exhibit similar spectral characteristics. Propydium iodide is excitable at non-UV wavelengths far more efficiently than its Ethidium bromide counterpart. This is desirable for laboratory use as UV wavelengths degrade DNA. A similar dye from a different family is acridine orange. This family of dyes is still very structurally similar to Ethidium bromide. Note the 3 fused aromatic rings at the core, particularly the presence of quinoline. Unlike the other dyes we've discussed, acridine orange is permeable to living cells. A desirable feature of acridine orange is its dual fluorescence. The dye is capable of staining both DNA and RNA, but by different mechanisms, hence giving different fluorescence. DNA fluoresces green, RNA fluoresces at a longer wavelength, giving a deep red color. This allows for distinguishing of the two nucleic acid types using a single dye. Another family of nucleic acid stains are the cyanide dyes. The most general structure includes two quaternary nitrogens joined by a conjugated polymethylene chain. The nitrogens are often members of independent heteroaromatic ring structures, some examples being quinoline, a feature of Ethidium bromide, and pearl. All cyanide dyes exhibit higher overall binding affinity relative to phenanthridine or acridine dyes. Spectral characteristics can be modified by modification of structure and substituents. All else being equal, spectral characteristics can be modified by increasing or decreasing the length of the polymethylene chain. For example, Psi3 connected by a 3-carbon chain fluoresces at shorter wavelengths than Psi5 connected by a 5-carbon chain. Cyanide dyes are preferred for many applications as they are capable of staining living cells, particularly the cyto family. These dyes generally have lower binding affinity, but are capable of passive diffusion through intact membranes. The structure of these dyes are proprietary, but can be seen in action here in this nuclear imaging. Like other cyanide dyes, the cyto family are customizable by color.
It is no surprise that one of the most common applications of Ethidium bromide is for agrostrial electrophoresis to visualize DNA. Another application is EMSA, which is short for electrophoretic mobility shift assay. This assay follows the same concept of agrostrial electrophoresis, except it is used to visualize protein DNA or protein RNA interactions. One application of propidium iodide is that it is used as a DNA stain for flow cytometry. Propidium iodide typically only stains dead cells, therefore it can differentiate live and dead cells, as seen on this picture. Hexidium iodide is a nucleic acid binding dye that selectively stains gram-positive organisms. The dye could help differentiate gram-positive bacteria from gram-negative bacteria. The picture on the left shows the dye being used to stain bacteria from a biofilm. Ethidium monoazide is a DNA stain that enters into bacteria with damaged membranes. A usage for this is to have the stain bind irreversibly to the DNA of dead bacteria. This could prevent PCR amplification of DNA from the dead bacteria. Shown on the diagram in the right, the dead cells can be amplified with PCR. An interesting application of acridine orange is for cell cycle studies because it emits a different color when bound to double-stranded DNA compared to a single-stranded DNA. When bound to double-stranded DNA, it emits a green fluorescence, while it emits a red fluorescence when it is bound to single-stranded DNA. Cyanide dye could be used as a fluorescence probe to label proteins or any type of biomolecules. Here are a few variations of cyanide dyes. Overall, Ethidium bromide and other intercalating dyes are important tools for studying cells and biomolecules. Thank you for watching our video.